nutrition a little bit, and urinary system. We'll be on this next exam. Again, acid-base will be on the last exam along with reproduction and whatever we do after that. But again, this next exam is 1 week from today, covering digestion through the urinary system. Any questions on the exam before we move on, I guess? If not, last time we were talking about acid-base balance, and we are going to continue with that today, and I fully expect we will finish with it. And start moving into reproduction, which is essentially the last bit we need to cover for this semester. And just remind ourselves what we're talking about when we're talking about an acid and a base. An acid is something that gives up hydrogen ion. A base is something that binds hydrogen ion. And we measure this in terms of pH, which is the negative log of the hydrogen ion concentration. Which simply means if we express the concentration as an exponent like this, we look at the exponent and take the negative of it. So if we have a concentration of 10 to the minus 7th, the pH would then be 7. If we had a negative hydrogen ion concentration of 10 to the minus 3, the pH would be 3, which would be a larger amount of hydrogen ion. Again, this is an inverse scale, and it is not at all linear. This is a logarithmic inverse. This is an inverse logarithmic scale. Again, anytime you have a pH less than 7, that means you have a high amount of hydrogen ion compared to a neutral, which means you are an acid. If you have a pH greater than 7, that means you're a base, and you have a low amount of hydrogen ion compared to neutral. Again, pure water has a pH of 7.0. Blood pH is more like 7.4. Blood pH is 7.4. All right, and again, when we're talking about acids and bases in the body, usually what we're trying to do is neutralize the acids, because we produce a lot of acids during our normal everyday metabolism. Every time we split ATP, we get an acid out of it, we get hydrogen ion out. So we're producing acids all the time. Some of these are said to be fixed in that you cannot breathe them out. Others are volatile in that you can breathe them out. And how we deal with these are different. The volatile ones, again, that main one being is carbonic acid, we breathe it out as carbon dioxide. We can use the respiratory system to eliminate that one. The fixed acids are acids that build up in the body. These have to be neutralized and then removed through the kidneys. So we have different mechanisms for dealing with these different kinds of acids. And again, this is important because protein folding is very much pH dependent. As are many of the physiological reactions are pH dependent as well. So it is very important that we maintain our pH in a very narrow range between 7.35 and 7.45. That is our normal arterial blood pH. You'll notice that this is arterial blood pH. The pH inside of your cells is going to be more neutral. Inside the veins, it could be more acidic. Depending if you have a very active system, say you're exercising heavily, you're going to be producing a lot more acid, and that can lower the pH inside of the veins. But the blood coming in should have that same pH of 7.4. So when we're talking about the pH for the rest of this, we're talking about blood pH, which again, normal range is 7.35 to 7.45. If you are below that 7.35, you are acidotic, or you have acidosis. If you're above 7.45, you're alkalinic, or have alkalosis again. And again, both of these are bad. Acidosis suppresses the central nervous system, leading things to not respond correctly because they're not getting those action potentials set off at the right amount, leading eventually to everything shutting down, coma and death. Alkalosis is the opposite. It makes the action potentials fire off too rapidly, and therefore you become hyperexcitable, and everything goes into contraction. You burn up all your ATP and you die. So again, both of them have the same results: death. If you let go too far out of the body. So again, we need to prevent us from getting too far away from that 7.35 to 7.45 range. And we have three mechanisms by which to do it. First of all, we have what are called buffers. Buffers are, again, weak acids and weak bases. The main one being carbonic acid. The carbonic acid bicarbonate buffer system is the primary buffer system that we're going to be using. And that is due to the fact that if we take carbonic acid, remove a hydrogen ion from it, that gives us bicarbonate, which can then bind that hydrogen again and keep us within that narrow range. We can also change our pH over time using our breathing. If our pH starts to drop, we can breathe out more of that carbon dioxide. Remember, carbon dioxide can be converted into carbonic acid and therefore acts like an acid. So the more we breathe, the more ventilation we have going on, the more carbon dioxide we are removing, and that will tend to push the pH up. That will tend to increase the pH by removing that carbonic acid. Likewise, if we decide to breathe less, we will retain more carbon dioxide and allow the pH to drop. The kidneys, of course, have the ability to add and subtract bicarbonate as needed. If our pH is getting too low, we add more bicarbonate in from the kidneys, and they will cause that will neutralize some of that acidity. If the pH is going too high, they can actively remove the bicarbonate. So again, we have buffers, ventilation, and renal effects, all of which work all the time, but with very different time scales. So again, these all work with very different time scales. Buffers are immediate, but very limited in their capacity because all you have is however much you have. So again, the buffers are immediate, but they're not actually removing the hydrogen ion, they're simply binding it. It has to be removed in a different way. Again, these are weak acids and bases, the main one being the bicarbonate carbonic acid buffer system. But this is not the only one, there are other ones out there as well. And again, it works based on this equation right here. Bicarbonate plus hydrogen ion forms carbonic acid. This can be converted into CO2 and water and breathed out. Or we can retain more carbon dioxide, convert it to this, and send it in this direction. So again, that is the primary buffer system here. At a pH of 7.4, this is in equilibrium. If something happens and adds more hydrogen ion here, it will tend to drive the reaction in this direction to get it back into equilibrium again. Likewise, if we remove the hydrogen ion, it will move it in the other direction to bring us back into equilibrium again. So again, that's how buffers are going to work. At equilibrium, we'll have a mixture here of both of them, and when they're in equal amounts, that's when we get a pH of 7.4. And this buffer system is what maintains your blood pH at 7.4. All right, so that's how the buffers are going to work. Again, we can add and subtract from either side of this system here. Again, if we're driving in this direction, we can then remove this by bringing it out as carbon dioxide. Likewise, we can add add bicarbonate onto this end in the form of sodium bicarbonate. Or we can do the opposite and drive in the other direction by breathing less, retaining more of this, and removing bicarbonate through the kidneys as well. Okay, so that's the bicarbonate buffer system. Again, we went over this last time. Ventilation works on the CO2 link. Again, CO2 plus hydrogen forms carbonic acid, which is again half of that bicarbonate buffer system. CO2 plus hydrogen ion forms carbonic acid, and this occurs in the bloodstream, specifically in the red blood cells, is where this is occurring. Using what is known as carbonic anhydrase. Carbonic anhydrase does this conversion, and it's located inside the red blood cells. But by breathing less, we retain more carbon dioxide, create more carbonic acid, lowers the pH. If we breathe out more, if we hyperventilate, we remove more carbon dioxide, thereby removing more carbonic acid. So again, ventilation works by adding and subtracting carbon dioxide. Adding and subtracting carbon dioxide. Again, it's not immediately needed, it does take some time, but you are actually removing the acid or adding the acid in the form of carbonic acid here. Again, decreasing breathing moves towards the right, increasing breathing moves it towards the left. I hope I got the structure correct. Anyway, the kidneys, however, have a much larger effect, but they're a lot slower. The kidneys are slow. They mainly work through changes in bicarbonates, mainly through bicarbonates. But they have basically an unlimited capacity. Mainly by manipulating bicarbonate levels. If we want to increase the pH, we add in more bicarbonate in the form of sodium bicarbonate. We increase bicarbonate from the kidneys, we reabsorb all of it, and we actually can make bicarbonate in the kidneys and the tubular cells. To decrease the pH, we excrete it. We can excrete it in large amounts. Which brings us to where we ended last time. If something happens and we become acidotic, our pH is dropping for some unknown reason. So what will our body do as a response to this? Well, first of all, the buffers are going to bind the hydrogen ion. Again, they can do this immediately, and for minute-to-minute changes in pH, this is what's going to be taking care of it for us. So if you decide you're going to hold your breath for a while, the buffers will be able to bind up that excess carbon dioxide. So again, this is going to be an immediate effect, very rapid, but very limited in its capacity. Once you bind up all the hydrogen, all the buffers, you can't do it anymore. So it is a limited capacity. And again, does not actually move into the acid. Longer term, we can change our ventilation rate. Again, this will work in the matter of minutes. We can change the patient using our ventilation in a matter of minutes. If we become acidotic, that means we can breathe out more carbon dioxide. In other words, we're going to increase our breathing rate. And by removing the carbon dioxide, we're removing the carbonic acid. Again, it's relatively fast. It can change the pH in a matter of minutes. Again, if you hold your breath for try to hold your breath for an extended period of time, we will be able to watch your pH drop. You can measure the pH. It does drop in a matter of minutes, leading you to want to breathe more. Again, this is a signal. But it removes the carbon dioxide and thereby increases pH through the removal of the carbonic acid. However, long term, we are going to increase renal reabsorption of bicarbonate, and we're going to create bicarbonate in the kidneys, but this is slow. It takes time to do this one. The kidneys are much slower than our breathing, but they have a much larger capacity to do this. So that is what's going to happen if something causes us to become acidotic. Again, as I've already mentioned, something happens, hydrogen ion goes up, we bind it with the buffers here, and we remove the carbonic acid by converting it into carbon dioxide and we blow it out. And we breathe it out. We add here the kidneys, we reabsorb as much of that bicarbonate as we can, so we then have it available to bind the hydrogen over here. And we can actually create sodium bicarbonate to bind even more of it. But again, this is a relatively slow process. But again, very high capacity, but slow. And hopefully this will all bring us back to normal relatively quickly. If instead we become alkalytic for some reason, you have a lot of diarrhea or something and you become alkalytic, we do the opposite. Actually, diarrhea is acidosis. If you're vomiting a lot, you get alkalytic. But anyway, the buffers will then release hydrogen ion, again, immediate, but limited capacity. Once you use it up, it's gone. We can decrease our ventilation, retain more carbon dioxide, and convert it into carbonic acid. Again, relatively fast, but only works on the one acid. And thereby decreasing the pH. And we can increase the excretion of bicarbonate. We can actually remove the bicarbonate, put it into the urine, and remove it from the body in that direction. Again, this is going to take hours and days to do. But can do as much as we want. And again, this is the previous figure, just working in the other direction. So again, something happens, we don't have enough hydrogen ion, we can breathe less, create more carbon dioxide in the body, convert it into carbonic acid, and increase the amount of hydrogen ion. we can remove the bicarbonate from this end, so it's not available to bind to that hydrogen ion, and we remove it through the kidneys as well. So again, very easy to change this. Okay. So, what can cause you to become acidic? Well, there are basically two ways of changing your blood pH and becoming more acidic. Number one is what is known as respiratory acidosis. In respiratory acidosis, the problem is you're not breathing enough. If you're holding your breath or otherwise unable to get enough ventilation going on, we retain too much carbon dioxide and our pH drops. That is respiratory acidosis. So in this case, the problem is we're not breathing enough to remove the excess carbon dioxide. You have some sort of COPD going on, or you're holding your breath, or your breathing rate is depressed because the central nervous system has become depressed for some reason. Something has happened and we're not breathing enough. That will cause our CO2 levels to rise, which will cause the pH to go down. Again, think of carbon dioxide as an acid. And that is known as respiratory acidosis. Anything else is called metabolic. Metabolic acidosis means we are breathing normally, but something else is causing us to causing us to retain too much acid. And again, so respiratory, the problem is we're not breathing enough. Metabolic, we're breathing just fine, but we're still seeing a drop in pH. And what could cause that? Well, there are a lot of things that can cause this. If you're exercising heavily, you produce a lot of lactic acid. That can cause the pH to drop. We could get ketone body production. When we get ketone body production, when we're burning fats. So again, when we burn fats, when you're starving. This is what is known as ketoacidosis. Ketoacidosis is because we're burning through our fats instead of using the carbohydrates that our body prefers. Again, this simply means either starvation and you're not getting enough calories, or you have diabetes and you're not actively using your carbohydrates correctly. Again, either of these will lead to ketone body production by burning the fats. Again, many of those ketone bodies are acids. We could have diarrhea. And during diarrhea, we lose a lot of bicarbonate through the feces because we're not reabsorbing it through the digestive system like we should be. So again, diarrhea can also lead to acidosis as well. But these are all metabolic causes. Again, you're breathing fine, but something else has happened and your pH is dropping for some other reason. Again, respiratory, the problem is you're not breathing enough. Metabolic, you're breathing fine, but something else is going wrong. Yes?
What would be the symptoms of somebody who has acidosis with metabolic?
Well, for one thing, and we'll get to it on the next slide. If you have metabolic acidosis, how are you going to compensate for that? You're going to breathe more. Your breathing rate will go up. That's one symptom. Your breathing rate increases to try to blow off the excess acid. Other things could be a depression of the central nervous system. Again, people could go into severe ketoacidosis. They become sluggish and just don't work as well. And eventually pass out and die if it gets bad.
Would your sweat smell a specific way if you were acidic?
Your sweat would not specifically smell, but your breath would. If you have ketoacidosis, your breath smells bad because of those ketone bodies. But yeah, it shouldn't cause any change in the way your sweat smells like. Because, well, first of all, your sweat doesn't smell. It's the bacteria that eats the sweat that smells. The sweat itself doesn't have any smell. It's salt water. It shouldn't be a difference in the sweat. But your breath could smell very different. Specifically ketoacidosis. Okay, so how do we respond to this? Well, if we have a respiratory acidosis, the problem is we're not breathing enough. If we're not breathing enough, how can we compensate for that? Well, again, the cause is the fact that we're not breathing enough. Ideally, you'd be able to increase your breathing rate. But again, that's the problem, is that we're not able to breathe enough. So we have to compensate in some other way. But again, this is going to cause a low pH, because again, acidosis by definition is a low pH. In metabolic acidosis, the problem is our pH is low, not because of the breathing, and therefore that low pH will lead to an increase in the breathing rate. So again, in respiratory, we're not breathing enough, and our pH is low. In metabolic, our pH is low, but our breathing has increased. Our breathing has increased to try and get rid of some of that acid. But again, so that means that our CO2 levels will also be low. In metabolic, we're going to have high levels of carbon dioxide. In metabolic, we're going to have low levels of carbon dioxide in the bloodstream. So you can easily distinguish between metabolic and respiratory by looking at the levels of CO2. If your CO2 is high, that means it is a respiratory problem. If your CO2 is low and the pH is low, that is a metabolic problem. So again, if you look at the pH and the carbon dioxide, if they're both elevated, that means, if they're both depressed, that's metabolic. If one is up and the other is down, that's respiratory. So again, in respiratory acidosis, how are we going to respond to this? By making more bicarbonate. Again, the problem here is that we're unable to get rid of the CO2 over here. Since we can't get rid of this, we have to do it from this end over here, and we're going to respond by producing more bicarbonate. By the way, CO2 and bicarbonate almost always go in the same direction. Those two almost always go in the same direction. So again, in respiratory acidosis, we're getting too much carbon dioxide building up, and it's going to lead us to have too much bicarbonate. And we're going to respond to that drop in pH by producing more bicarbonate to try and get more bicarbonate. All right. So again, respiratory acidosis, something happens, we can't breathe enough, leading to an increase in CO2 levels, leading to an increase in carbonic acid, leading us to have a lowered pH. To respond to that, we're going to try and change our breathing rate if we can. Again, this is a problem, so that's probably not going to happen too well. And we're mainly going to be dependent on the kidneys to get rid of that excess acid by adding in more bicarbonate. We're going to increase secretion of bicarbonate. By doing that, our pH will then drop back down to normal. That is the response to a respiratory acidosis. Main thing is we're going to increase the amount of bicarbonate to try and compensate for it. In metabolic acidosis, we're able to compensate using the lungs by blowing off that excess CO2. So in metabolic acidosis, we're going to see a drop in pH because something happened and our pH has dropped. And we're going to, for instance, we're producing too much for some reason, we're exercising or whatever. Or we're, for some reason, not able to produce enough bicarbonate. Those are the two things. But anyways, in metabolic acidosis, we're going to see a drop in pH, not because of the carbon dioxide. So to respond to that, we're going to change our breathing rate. So again, something happens, our pH levels have dropped, which means we have increased hydrogen ion, which means we're going to, number one, change our breathing rate. And we're going to increase our breathing rate, blow off the excess CO2. As the CO2 levels drop, our carbon dioxide levels drop, our carbonic acid levels drop, which means our bicarbonate levels drop. So in this case, we're going to see a drop here, mainly due to the change in CO2 levels. And CO2 levels are going to be dropping and destroyed. So again, that's one way of doing this. This is another way of doing the same thing. But this is from the bicarbonate side. Alkalosis, basically the opposite. Basically we do the opposite. Again, it could be respiratory or it could be metabolic. Again, respiratory alkalosis means we're breathing too much, we're hyperventilating. And we're getting too much carbon dioxide being lost. Again, this one is not that common. But if you're breathing too rapidly, you saw a UFO and you started breathing really rapidly, we could see an increase in your blood pH as a result of that. Or it could be metabolic. Most common cause, vomiting. Because as you're vomiting, you're removing all that stomach acid, and that's lowering the pH in the bloodstream as well to replace it. So again, could be something else, but not in here as well. By the way, acidosis is far more common than alkalosis. Most of the time, it's acidosis is the issue, not alkalosis. But anyway, how do we respond to this? Well, it's respiratory. Slow down your breathing rate. But again, the problem here is the fact that we're breathing too rapidly, causing our CO2 levels to drop, and for some reason we're unable to retain that CO2. Again, this is going to lead to a lowering in the CO2 levels and a lowering in the bicarbonate levels. So in the respiratory alkalosis, we're breathing rapidly, our pH is dropping, and our CO2 levels are dropping as well. They're going in the same direction. So we're going to see high pH, low CO2. In metabolic, this is going to be the opposite. We're going to see high levels of pH because we're losing the acid, and we're going to see high levels of CO2 as well. Because we're trying to compensate for by changing our breathing rate and coming down our breathing rate. Again, look at those previous figures, run them in the other direction back into this. So again, in respiratory alkalosis, we're hyperventilating. That lower CO2 levels leads to an increase in the pH because we're removing excess carbonic acid. The easiest way to deal with this is to slow down your breathing rate. But if we're unable to do that, we're going to remove bicarbonate. We're going to remove bicarbonate to get rid of it. And again, that will bring our pH back down. That will bring our pH back down. Yeah, down. Bring our pH back down. Metabolic, same idea. Again, number one way is we're going to change our breathing rate. Causing our CO2 levels to go down and leading to we're going to slow down our breathing rate, causing the CO2 levels to go up as the pH comes down. Okay, and all of this can be summarized in a handy-dandy little table here. So respiratory acidosis, the problem is we're breathing too little and our CO2 levels are going up. Therefore, increasing the amount of carbonic acid and therefore increasing the amount of bicarbonate. So we're going to see high CO2, low pH. So in respiratory acidosis, we have high CO2 and low pH. In respiratory alkalosis, it's the opposite. We're breathing too much and our CO2 levels are going down, which is causing our carbonic acid levels to go down, which means our bicarbonate levels are going down as well, but our pH is going high. And there's a easy way to remember this. Take a look at the CO2 levels and the pH here in respiratory acidosis or alkalosis. The CO2 and the pH are going in opposite directions. So, I'm sorry, they're going in the same direction. Increased CO2 leads to increased, that's very opposite. But anyway, the CO2 levels are going to be high, our pH is going to be low in respiratory. They go in opposite directions. pH drops and the CO2 goes high. Over here, pH goes high, CO2 goes low. CO2 goes high and pH goes low. All right, metabolic, alkalosis, they go in the same direction. pH goes, the pH goes low in acidosis, and our CO2 goes low as a result. Likewise here, increase of pH, increase CO2. By the way, there's a nice mnemonic to remember this by. Again, when we're looking at the CO2 levels and comparing the CO2 and the pH levels in respiratory, they are opposite. In metabolic, they're equal. They go in the same direction. So comparing the CO2 and the pH levels, if they go in opposite direction, it is a respiratory problem. If the pH is dropping, it's acidosis. If the pH is rising, it's alkalosis. In metabolic, they go in the same direction. If the pH levels are dropping, the carbon dioxide levels are dropping. So again, looking at carbon dioxide and pH, if they're going in the same direction, it's metabolic. If they're going in opposite directions, it is respiratory. And again, anytime the pH is high, that's alkalosis. Anytime the pH is low, that is acidosis. Is that, I'm seeing confused looks out there. Yes?
Can you just give us like a question that you might ask?
Oh, on an exam.
The actual.
A kind of question that you might see, a patient comes in, and they have high levels of, they're showing the symptoms of acidosis. You take a look at their blood gases and you find the carbon dioxide levels are high with a low pH. Is that a respiratory problem or a metabolic problem?
Respiratory.
Respiratory. That's the kind of question you might get.
Okay.
How about one more?
All right, a patient comes in after seeing a UFO, they've been hyperventilating, and they're showing the effects of that, and you take a look at their blood gases, you find their CO2 levels are very low, and their blood pH is very high. Is that respiratory or metabolic?
Respiratory.
That's also respiratory. Is it alkalosis or acidosis?
Alcohol.
Alkalosis. Yep. Again, if the pH is high, it's alkalosis. Whether or not if it's because of the carbon dioxide, it's respiratory. If it's not, if it's something else, it's metabolic. Okay?
Mm-hmm.
Okay. And that's basically acid-base balance in a nutshell. Again, we could spend a lot more time on this, but we're not ready. Any questions on acid-base balance? And again, it's mainly having to do with the respiratory system and the kidneys that are regulated. All right, in that case, we now need to move on to the last major topic of the semester, and that's reproduction, making more people. And again, when we're talking about the reproductive system, obviously it is different male and female. We're going to start with the male reproductive system. And in the male reproductive, the anatomy is a little more complicated, but the physiology is a lot easier. In the female, the anatomy is fairly simple, but the hormones are horrible. Again, the physiology behind it is going to be very complicated. So again, be prepared for this. But in both cases, we need to talk about number one, the gonads. And what are the gonads? In the male, they are the testes. In the female, they are the ovaries. And they are responsible for creating the, what is known as the gametes. What are gametes? Eggs and sperm. Those are known as the gametes. Again, the ovaries produce the eggs. The testes produce the sperm. Again, you learned about this. I'm hoping you learned this in the biology class. But the main function of the gonads is to produce the gametes. They also produce a number of hormones. Again, those gametes, the gonads also produce a lot of hormones. Specifically, the testes produce a lot of testosterone, and the ovaries produce a lot of estrogen and progesterone. In other words, your same-sex hormones are coming predominantly from the gonads. We all have both sets of hormones, at least we should have. You get your opposite sex hormones from the adrenal glands, but in low amounts. The adrenal glands produce a little bit of testosterone. The testes produce a lot of testosterone. Likewise, the adrenal glands produce a little estrogen. The ovaries produce a lot of estrogen. So again, what makes the difference between looking male and looking female isn't the hormone, but how much of it do you have. Again, it is the relative amount that determine whether you are male or female in appearance. In addition to the gonads, we also have a bunch of ducts involved. And the ducts are there to transport the gametes from where they're being produced, from the gonads, to wherever it is that they need to end up. In the male, that means taking the sperm from the testes and depositing them into the female reproductive tract. In the female, it means taking that egg and getting it first into the uterus and then out of the body after the end of gestation. So we'll have a number of ducts involved as well. There are a number of accessory glands there to make sure everything goes smoothly by producing a number of different kinds of fluids, lubricants and other kinds of fluids to make sure that everything goes the way we want it to work. And there will be a bunch of what are known as perineal structures, also known as the external genitalia. These perineal structures, by the way, embryologically, they start out as indifferent. And then they become either the male structures in the presence of testosterone, or they become the female structures in the absence of testosterone. So again, they started out as a mass of cells, and their developmental fate is dependent on whether or not you have a Y chromosome, essentially. And we'll talk more about that in a few more slides. So again, these, for both male and females, we'll be talking about gonads, ducts, accessory glands, and perineal structures. Again, all involved in making sure that we end up with so first thing we need to consider is what determines whether you're going to be male or female. And by the way, when I'm talking about male and female, I'm talking about genetically and phenotypically male and female. I'm not talking about gender here, I'm talking about biology in all of this. So which sets of structures develop is dependent on whether or not you have something called TDF, testes determining factor. Testes determining factor is a gene that is found on the Y chromosome. Again, in biology class, you should have learned that men are XY, women are XX. Only men have a Y chromosome, which means only men will produce TDF. And this is the thing that is going to determine whether you're going to end up being male or if you're going to end up being female, is whether or not you have TDF. So again, if you don't have a Y chromosome, you will not produce TDF because that gene doesn't exist in you. All right, so again, that TDF is found on the Y chromosome. Only men get the Y chromosome. Only men get it. By the way, when we're talking about the Y chromosome is one of the smallest of the chromosomes. It basically has things to turn you male on it. And that's basically all that is on that chromosome. The X chromosome is has a lot of other stuff on it as well, like your code of pigments are on the X chromosome. So again, the Y chromosome is basically just to turn you male, and the X chromosome does other things too. So again, you want to have both an X and you need to have an X, and the Y is optional. Okay, in the absence of TDF, you turn female. Or if, again, there are people out there who have a Y chromosome but the TDF gene is not working, they're phenotypically female, even though they have a Y chromosome. If you don't have TDF, a functional TDF gene, you are female. Female is the default form in humans. In humans, female is the default form. You get female because you actively turn that system on, otherwise you are female. Otherwise you're a female. The male ducts will derive from what is known as the Wolfram duct system, also called the mesonephric duct system. The female ducts derive from what are called the Mullerian duct system, or the paramesonephric duct system. By the way, you'll notice both of these have nephric in them. We've seen this term before. What did it refer to?
The nephron?
Which is where?
In the kidney.
In the kidneys. Why this is called mesonephric and paramesonephric is because these are by the kidneys. Developmentally, everything starts up by the kidneys. It does not descend into the pelvis until late in pregnancy. Late in gestation is when all these things happen. It's not the final position. But again, the male ducts derive from the Wolfing ducts, the female ducts derive from the Mullerian ducts. Initially, you have both. We start out by having both of these. We start out with both of these. And then you actively turn one on and turn the other one off, depending on whether or not you have testosterone around. Again, the key to all of this is going to be testosterone. The both ducts are present in the embryo we have both. So, in the presence of TDF, that's the determining factor. What is known as the indifferent gonad, again, we start out, the testes and the ovaries start out as the same structure. They start out as the same structure. And they will either turn into one or the other, dependent on whether or not you have TDF. If you have TDF, that indifferent gonad, which is up by the kidneys, turns into testes and you become male. If you don't have TDF, it turns into an ovary and you become female as a result. So again, the testes develop in the presence of TDF, the testes develop, and they then start producing testosterone and MIF. MIF is Mullerian inhibition factor. So again, this occurs early on during pregnancy, or this occurs early on embryonically. This happens within the first few weeks of pregnancy. The determination whether to be male or female happens very early. And by the way, we see a large increase in testosterone in that early embryo. In males, we see a large increase in testosterone. If you time it right, you can actually measure that in the female, in the mother's bloodstream as well. Unfortunately, this is before most people realize they're pregnant. So again, we do see that big surge in testosterone to turn you male fairly early on, within about a month of implantation. All right. So what's that testosterone do? Well, it leads to development of the male structures, including that Wolfing duct system, causes it to develop into the epididymis, the ductus deferens, the ejaculatory ducts. Those are the male ducts. And we'll talk about where those are at a little bit later on. And those different structures are going to turn into the penis, the scrotum, and the prostate. Those are the perinatal structures in the male. The MIF turns off the Mullerian duct system. It turns off the female structures. So again, we see degeneration of the Mullerian ducts due to the MIF being produced. And again, this is in the presence of TDF. Now, occasionally you will run across an individual who has the TDF, but it doesn't produce the testosterone or the MIF correctly, and then you end up with somebody who, at birth, it is unclear whether they're trying to be male or female. And that is what is known as intersex, which happens a lot more frequently than people realize. But again, you could end up with what are called indifferent genitalia if not everything works according to plan. Most of the time it does. But sometimes things go wrong. In the absence of TDF, you get ovaries instead. You don't develop testes, you develop ovaries. And no MIF or testosterone is being produced because you don't have testes. And that leads to development of the Mullerian ducts because we're not turning it off. So the Mullerian duct system develops into the uterus and the uterine tubes. Those are the female ducts, uterus and uterine tubes. The Wolfing duct system then degenerates because it's not being supported by the testosterone. In the absence of testosterone, we turn off the Wolfing ducts. And the other embryonic structures develop into the labia, the vagina, and the clitoris. Will develop in the female instead. So again, in the presence of TDF, you get the male system and you turn off the female system. In the absence of TDF, you develop the female system and the male system does not develop. Okay. So let's say we have determined that we are going to have a Y chromosome with a functional TDF gene, and we are going to turn into a male. We will develop testes, which are then the gonads. Again, this happens quite early on, and those testes will secrete what are known as the androgens. The androgens are the male hormones. The androgens are the male hormones, and the main one is testosterone. Testosterone does come in several different forms, but for the purposes of this class, I'm just going to say testosterone. Be aware there's more than one kind. Likewise, I'm just going to say estrogen. There's more than one kind. There are multiple forms of both testosterone and estrogen. I'm just going to lump them all together as testosterone and estrogen. So again, the testes develop and they secrete the male hormones, the main one being testosterone. It also will then produce a sperm at the beginning of puberty. We'll start getting sperm production. The ducts will then develop from the Mullerian duct system into the epididymis, the ductus deferens, the ejaculatory duct, and the urethra. In the male, the urethra pulls double duty. It is both a urinary structure and a reproductive structure. In the female, it is only a urinary structure. And the reproductive structures are completely separate. But in the male, it enters into the urethra. The epididymis is a very coiled up tube that sits around the back of the testes, and this is where final sperm maturation occurs. The ductus deferens is then there to carry the sperm from the epididymis up towards the ejaculatory duct, which runs through the prostate gland. The ejaculatory duct then mixes is where the mixing of the sperm with the semen occurs in the ejaculatory duct, and the whole thing then comes out through the urethra on the way out. So that's in reproductive, and we'll look at it more later. The accessory organs are then the seminal vesicles, the prostate, and the bulbourethral glands. The seminal vesicles produce most of the semen, about 70% of the volume of the semen comes from the seminal vesicles. The prostate gland adds another 10-15% of fluid into the semen as well. And between the two of these, we'll get the semen being produced. And we'll talk about why we need these two separate glands a little bit later on. The bulbourethral glands will then secrete a fluid, which is essentially a lubricant, so that the sperm can come out more easily. So the bulbourethral glands are producing lubricant as well as a lot of bicarbonate. As it turns out, sperm does not like acid, and the female reproductive tract tends to be fairly acidic. So again, we've got bulbourethral glands. The semen contains a lot of bicarbonate to neutralize the acids in there. And the bulbourethral glands are producing lubricant and helping to neutralize some of that and clean it, flush it out. And then we have the external genitalia, which in the male consists of the penis and the scrotum. The penis is to allow copulation to occur, and the scrotum is there to regulate the temperature of the testes. As it turns out, to get good sperm production, you want the temperature to be a bit lower than the internal body temperature. So instead of being at 37 degrees, you want to be more like 34 degrees. So again, we want to keep the testes on the cool side for optimum sperm production. So what does all this look like? Well, it looks something like this. First of all, here are the testes down here, and you see there's this flap of skin coming around here. That is the scrotum. That's the scrotum. So the testes are contained within the scrotum, and where it is hanging out of the body cavity where it's a little bit cooler. To get good sperm production occurring, again, we want to keep a little bit cooler. And the testes in here, we'll spend some time talking about them. They're egg-shaped, they're light as you can see here, and they're about yay big, the size of a small egg. Few centimeters. On the back of them, you see this sort of comma-shaped structure right here. That's the epididymis. The testes make the sperm, and upon being produced, they are then stored in the epididymis. So the sperm are then stored in the epididymis, which is simply a very long coiled tube. If we were to take that epididymis and stretch it out, it's about 6 feet long. It's about 6 feet long. So again, the epididymis is a very tightly coiled tube which runs along the back of the testes. And again, we store our sperm there until ready for use, and sperm undergoes its final maturation here in the epididymis. As the sperm comes out of the testes, they are not quite ready for use yet. They do exit in the epididymis for a little bit before they become active. By the way, men produce on the order of 1 to 300 million sperm per day. A normal healthy adult male will produce on the order of 100 million to 300 million sperm per day. To put that number into context, that's enough sperm to impregnate every female in North America on a daily basis. Men have a very large reproductive capacity. In contrast to that, women are born with about 1 million eggs, and that's all they'll ever have. And one will be released roughly once a month. Roughly about 400 eggs will be produced during a woman's reproductive lifespan. Will produce about 400 mature eggs. So there's a very much difference in the reproductive capacity, male versus female. Women have a much lower reproductive capacity. All right, then you'll notice there's this tube coming out of the epididymis. That is the ductus deferens. You may have learned this one as the vas deferens instead. These days we're calling it the ductus deferens. The ductus deferens and the vas deferens are the same thing. And it's this tube here that carries the sperm from the epididymis up into the bladder, up into the body, around, behind the bladder, and then back down here into this structure right here, which is the prostate. And it's going to come back here on the backside. The end of it here, you see there's a little bit swollen there. That is called the ampulla of the ductus deferens. An ampulla is always a wide section. So there's a wide section at the end, and the sperm will gather there before being released from the body. The sperm will gather there with appropriate stimulation. It then enters into this tube right here, and this tube is the ejaculatory duct. That tube is what is known as the ejaculatory duct. The ejaculatory duct, you'll notice, is crossing through this gland right underneath the bladder, and that is the prostate. So this is the prostate. And the ejaculatory duct is crossing the prostate and mixing with the semen. So the semen is coming from these two structures right here. That will come back to a little bit later. That's the prostate and the seminal vesicles. They're producing the semen, mixing it with the sperm inside the ejaculatory duct. The ejaculatory duct then enters into what is known as the prostatic urethra. Again, we've already seen the urethra. It's what's draining the bladder here. But you'll notice the ejaculatory duct goes into the prostatic urethra here. And from there, into the membranous urethra, and through the spongy urethra and out the body. So again, that is the pathway that sperm are going to make. Once again, they're going to start within the testes. That's where the stem cells for making sperm are located. And that is where they will undergo their physical maturation. And we'll talk a lot more about that, probably not today. But again, the testes, specifically what are known as the seminiferous tubules, is where the sperm are being produced. And again, a lot of sperm are being produced on a daily basis. Upon reaching physical maturation, they then detach from the tubules, make their way out of the testes, and are then stored in the epididymis. They're then stored within the epididymis. And they'll undergo their final functional maturation in the epididymis. Again, going from that initial stem cell to mature sperm takes about 2 months. It takes about 2 months to make a sperm. But again, once that occurs, they will then be stored in the epididymis until appropriate stimulation occurs. And then leave it to your imagination as to what appropriate stimulation is. From there, they will then be transported through the ductus deferens, also known as the vas deferens, to the ejaculatory duct, where they will mix with the fluids being produced by the prostate and the seminal vesicles. Again, those fluids are known as semen. So the sperm will mix with the semen within the ejaculatory duct. and then be transported out the body through the urethra. By the way, all this movement is being done by peristalsis. These structures are muscular tubes, and it's peristaltic contractions that's pushing things along. All right, and that is the pathway that sperm take. We'll be adding more details into this shortly. So again, that's the pathway that the sperm take. We then have a number of accessory organs as well. One of the major ones is hanging out right behind the bladder here, and these are what are known as the seminal vesicles. So the seminal vesicles are hanging out here right behind the bladder. They're hanging out here behind the bladder. Those are the seminal vesicles. And they are producing most of the semen. They're producing most of the semen. Also, we have the prostate. The prostate is then producing another 10, 15, 20% of the semen. It's coming from the prostate, which is a roughly walnut-sized gland right underneath the bladder. Once again, we've seen this gland before. Now, one of the problems that men have as men age is the prostate has a tendency to enlarge and get bigger. And as the prostate enlarges, it can cause problems with these tubes that are running through it. By the way, that enlargement could be benign or it could be malignant. Prostate cancer is actually one of the more common cancers, and only half the population has prostate. But prostate cancer, most of the time, is fairly slow growing, and you'll die of something else first. But some forms of it are more aggressive and have to be dealt with. And to keep track of what is going on with the prostate, they will do what is known as a digital examination. And that involves feeling the prostate, because as it turns out, it's right next to this structure right here. What's this? That's the rectum. That's the rectum. So by inserting a finger up here, you can feel the prostate. Again, that typically begins around age 40 or so. This is why men don't like going to the doctor. But it's not the reason. It's not as bad as it sounds. That's what the prostate is. It's easy to reach. So it's very easy to keep track of what's going on with the prostate here. And if you see it beginning to enlarge, that can lead to problems with both the urinary system and the reproductive system, and it will be malignant. So again, that's what the prostate is. All right, we then have one other gland down here, and that is the bulbourethral gland. The bulbourethral gland is this little gland hanging out right here. It's called the bulbourethral gland because this right here, this is the bulb of the penis. This is the bulb of the penis. So it's between the bulb of the penis and the urethra. Hence bulbourethral. This was formerly known as Cowper's gland. This was formerly known as Cowper's gland. And the main function of the bulbourethral gland is to produce a lubricant. It produces a lubricant. Which, number one, makes all of this a little more slippery. And number two, helps to neutralize any of the acids that may be within the urethra. That is what the bulbourethral gland is doing. So again, the seminal vesicles, the prostate, and the bulbourethral glands all produce fluids, which are then mixed in with the sperm as it makes its way out the body. Then we have a couple of other structures here. The most obvious one is obviously the penis. By the way, the penis is there to allow penetration into the female reproductive tract, to allow the sperm to get into the female reproductive tract. And you'll notice the penis goes way back to here. About a third of the penis is internal. It's actually attached to the ischotuberosities on the backside. But again, that's the penis, and we'll talk about how the penis is put together a little bit later. We'll also notice that we have this flap of skin, again, I already mentioned it, called the scrotum, and that is there to regulate the temperature of the testes. Again, the scrotum is basically a flap of skin that we keep the testes in. There are a couple of muscles in here to allow us to pull up and release here on the testes to help to regulate the temperature. They are temperature-sensitive muscles, so that when it's cold, they shrivel up, bring the testes up closer. When it's hot out, they allow it to hang farther down. And that's the scrotum hanging down there. All right. So that's the basic anatomy here. let's take a closer look at the testes. The testes consist of two parts. 90% of it, of the volume there, is what is known as the seminiferous tubules. The seminiferous tubules is where the sperm is actually produced. And these are tubes. Each testis contains about half a mile of these tubes. In other words, there's a lot of space in here. We've got about half a mile in each testis, or put both of them together, you got about a mile's worth of seminiferous tubules. Each individual tubule is about 80 centimeters long, about like that. So if you stretch them out, they'd be about this long. And again, they're obviously very tightly coiled up to fit within the testes. And again, this is the site of sperm production, and the reason men have so many of them is because men need to make a lot of sperm. Again, I said men make a couple 100 million sperm per day. The vast majority of those sperm will never make it anywhere as close to an egg. Under optimal conditions, you might get 20 out to where the egg actually ends up. Most of them will never get anywhere as close to an egg. There's a reason men need to produce this many sperm. Inside those seminiferous tubules, we have what are called Sertoli cells. Your book is calling these nurse cells. These are also known as nurse cells. And those Sertoli cells are there to guide the development of the sperm. They're helping to guide the development of the sperm as they're being produced. So they're supporting spermatogenesis. Spermatogenesis is a fancy word that means sperm production. So again, the Sertoli cells, they're producing a couple of enzymes, not enzymes, they're producing a couple of hormones helping to regulate all of this. In addition to these seminiferous tubules, the remaining 10% is known as interstitial tissue. And this is what's surrounding the seminiferous tubules, and this contains what are called Leydig cells. These are producing the testosterone. So the testosterone is coming from the Leydig cells outside of the tubules. So again, the portion of the testes that produces the testosterone is outside of the seminiferous tubules in what are called the Leydig cells. This is basically connective tissue surrounding it, and it has these cells inside of it called Leydig cells. And it looks something like this. So here we see the seminiferous tubules hanging out here. Lots and very highly coiled up tubes hanging out here inside of here, and there's a whole bunch of them. The whole thing is then surrounded by a nice membrane on the outside called the tunica albuginea, which will be on another slide. But we have this nice white covering on the outside called the tunica albuginea, by the way. The ovaries started as the same structure. They also have a tunica albuginea, which simply means white covering. But these are the seminiferous tubules in here, and in cross section they look something like this. They look like this. And you can see here we've got our spermatogonia, which are the developing the sperm cells. I'm sorry. The spermatogonia are the stem cells that lead to sperm production. And they're out here towards the outside. And as they develop, they move towards the middle of the tubule, and then as they become structurally mature, they then detach and make their way out. So let's take a little closer look at what these tubules look like. Once again, if we blow it all up, we can see here's the edge of our seminiferous tubules out here. Down outside here, we have the interstitial space out here, and these are the Leydig cells out here that are going to be producing the testosterone. So the testosterone is produced by these Leydig cells out here. The spermatogonia are on the outer portion of the seminiferous tubules, and these are the stem cells for new sperm production. Again, we'll spend quite a bit of time talking about spermatogenesis. But it starts at the spermatogonia, and they're constantly dividing and repopulating themselves. So these are constantly undergoing mitosis and dividing. And then some of them are going to start to then differentiate into mature sperm over here. And as they go from here to here, they are aided along by these larger cells out here. These are known as the Sertoli cells. also known as the nerve cells. These are the nerve cells. So the nerve cells here are these big cells that you see in between the developing sperm, the spermatocytes. So again, out here we have the spermatogonia. Up here we have the developing spermatocytes. And then we have these nice big nerve cells known as Sertoli cells. One other thing to mention is that there is what is known as a blood-testes barrier. The sperm are technically not part of the person producing them. And the immune system, if it was able to get in here, would be attacking them. Because they have cell markers on them that are different from the cell markers of the person producing them. So again, we have what is called a blood-testes barrier. This is similar to the blood-brain barrier that we saw in the central nervous system. We don't let the blood get into here. And that blood-brain barrier is caused by these nerve cells. There are tight junctions between the nerve cells here that prevent fluid from seeping into here to keep the immune system out. So again, to prevent our immune system from attacking the sperm cells, we institute a blood-testes barrier to keep the immune system from attacking the sperm cells. So again, there is a blood-testes barrier here as well. All right, so technically when we get up into here, we're outside the body. All right, so that is the seminiferous tubules, what they look like roughly. And again, there's about half a mile of these in each testes. The whole thing is then covered by a membrane known as the tunica albuginea. Albuginea simply means white. It's a nice white covering on the outside, a nice connective tissue layer on the outside. There's a similar structure on the ovaries. Because again, ovaries and testes started out as the same thing. And it has a nice white covering on the outside. That is the tunica albuginea, surrounding it here on the outside. Tunica simply means a covering. So this is the tunica albuginea, and you can see all the seminiferous tubules hanging out here on the inside. In addition to that, so again, we have those seminiferous tubules hanging out here on the inside. And here you can see the testes sitting inside the scrotum hanging out here. And you'll notice on the outside, we do have a serous membrane on the outside here as well. There is a serous membrane surrounding the testes, and that is known as the tunica vaginalis. That is the tunica vaginalis, is the serous membrane surrounding the testes. It sounds like it should be a female structure, it is a male structure, the tunica vaginalis. And we'll talk more about that when we talk about the scrotum. But you'll notice the scrotum has two chambers on the inside. The two testes are kept in separate chambers on the inside. Oh, I forgot to mention something. Back with the blood-testes barrier. If something happens and you damage the testes, usually you end up losing the testes because the immune system attacks it. And will attempt to get rid of it. If men undergo some sort of a crushing injury in there where it damages the testes, usually you have to have it removed. So that you don't lose both. It's better to have one than the other. So again, anything that breaks this barrier can lead to the immune system attacking the testes. All right, that's something to learn. Okay, those are the seminiferous tubules, and you can see each one is contained within its own little chamber. Then, coming out of the seminiferous tubules, we have what are known as spray tubules, or tubuli recti in Latin. Tubuli recti means straight. The tubuli recti, or the straight tubules, are then leading from the seminiferous tubules into this network of little tubes up in here called the ready testes. That is what is known as the ready testes. The ready testes is sort of a mesh of, or a network of little tubules where the sperm will be collected before leaving the testes. They will then exit the testes through these little ducts right here, which are known as efferent ductules. The efferent ductules then carry the sperm from the ready testes out and into the epididymis. All right, so one more time. Within the testes, again, the whole thing is surrounded by the tunica albuginea. Then we have these long coiled up tubes here called seminiferous tubules. They are drained out by the straight tubules into the ready testes, which is then drained out by the efferent ductus into this big long coiled tube called the epididymis. So that is the pathway that the testes will make, starting from the beginning inside the seminiferous tubules out into the epididymis. Again, the epididymis here is, as you can see, a very long coiled tube. And it runs along the back of the testes. This runs alongside the back side of the testes. It has a head here at the top. Most of it's the body, and then a tail portion down here at the bottom. So it does have a head, a body, and a tail, but it's really just a coiled up tube. It is a very, very coiled up tube. Again, stretched out, it's about 6 feet long. It's about 6 feet long. And the sperm will be stored within the epididymis until ready for use. And it will undergo its final functional maturation within the epididymis. Again, as the sperm leaves the testes, it is not quite fully mature yet. It is structurally, it looks fine, but it doesn't quite work yet. It has to hang out in the epididymis for a while before it becomes functionally mature. Final maturation occurs in the epididymis. From there, we then exit using the ductus deferens, which is this tube right here. So this tube is the ductus deferens, and it goes up and into the abdominal cavity. It goes up into the abdomen. And it passes through what is known as the spermatic cord. The spermatic cord is a sort of a rope that is suspending the testes within the sperm. And inside that spermatic cord, we have the ductus deferens, the testicular artery, and the testicular veins. So again, the testicular arteries and veins are running through that spermatic cord as well, along with a bunch of connective tissue holding the whole thing together. So there are basically a number of tubes running through the spermatic cord, and again, this is suspending the testes within the scrotum, and it contains the ductus deferens, carrying the sperm up into the body, the testicular artery carrying the blood to the testes, and the testicular veins draining the blood back out. All run through the spermatic cord. We will talk about where the spermatic cord goes a little bit later on. But the spermatic cord runs through the inguinal ligaments. It runs through the inguinal ligament, as we noted what that is previously. From there, we continue up into the body, and we wrap around onto the back of the bladder. We wrap around here onto the back of the bladder. So again, here we see the urinary bladder. These are the ureters coming down from the kidneys, and these are the seminiferous tubules coming in back here. These are the seminiferous tubules. So again, there's the bladder, the ureter, and the ductus deferens coming down back to here. The ductus deferens here, you'll notice the end of it is swollen. We have this wide section here. That is known as the ampulla of the ductus deferens. Again, an ampulla is always a wide section. The ampulla is always a wide section. And we have this nice widened portion here, and the sperm will gather here before the final push out of the body. They'll gather here before the final push out of the body. In the ampulla of the ductus deferens. At the end of the ampulla, we have the output of the seminal vesicles. The seminal vesicles are these little sort of balloon-shaped structures here on the back of the bladder. And again, their function is to produce semen. Again, most of the fluids of the semen, about 70-75% of it, is coming from the seminal vesicles. And we'll talk more about what the semen is there for, but it's basically there to keep the sperm happy and healthy on its way trying to find an egg. So again, we'll talk more about sperm later on. From there, we enter into the ejaculatory duct. The ejaculatory duct then passes through the prostate, and again, here's the prostate hanging out here. So again, you can see here that the output of the ductus deferens and the output of the seminal vesicles join together here within the ejaculatory duct. And the ejaculatory duct will also receive the prostatic fluids as well. As it passes through the prostate, it will get fluids from the prostate, including things like prostaglandin and clotting factors, which are coming from the prostate. And we'll talk more about that later on. The prostate is this gland just a little bit bladder. And again, you can see the bulbar glands there at the bottom of this structure. So again, how does the sperm make their way out of the body? Again, they begin their life inside the seminiferous tubules as spermatogonia towards the edges of the seminiferous tubules. From there, they will grow and develop, and when they become structurally mature, they will release from the seminiferous tubules and go into the tubuli recti, or the straight tubules, and enter into the ready testes, that sort of a network of tubes towards the top of the testes. From there, they will then exit the testes through the efferent ductules. All of these are inside the testes. These are all in internal structures of the testes. Again, this is all inside the testes. Is that a question?
What was that, where they gather inside the ductus deferens before they go out, what is it called, how do you spell that?
Ampulla.
Ampulla, okay.
So again, all of these structures are inside the testes itself. We then leave the efferent ductules and enter into the epididymis, where these sperm are stored and undergo their final functional maturation. From there, upon appropriate stimulation, they will enter into the ductus deferens, also known as the vas deferens, where they will then enter into the ejaculatory duct, along with the outputs from the seminal vesicles. They'll also get the inputs from the prostate at this point as well. They'll then cross through the prostate, cross into the prostatic urethra, into the membranous urethra, and into the spongy urethra, where they will get the inputs from the bulbal urethra glands. We'll come in here as well. Yes, you should know this order. You should know this pathway. Yes, you should know this pathway. But that is the pathway that sperm take during their development until they leave the body through the penis. Yes.
Would you say 5 through 10 is outside the testes then?
Yes.
Okay.
Yes. So the epididymis is a tube along the back of the testes where the sperm are stored. The ductus deferens is there to take the testes up into where they will combine with the ejaculatory. All right, so that is the pathway of the sperm making its way through. We've got about 2 minutes left. I think this is a good stopping point. Next time we'll talk about the other